All right, so let's go ahead and get started. So make sure you have your review packet. And here we go. All right, so I did see in the chat someone answered this first question, which is great. And so enzymes are going to be proteins, right? They're actually catalytic proteins. And then what are proteins made out of? What are the building blocks of proteins called? This is kind of going back to one of the earlier lab exercises that you had. Right, yeah, so proteins are made out of amino acids. So anything that's made of a protein, like enzymes, are going to be constructed out of amino acids, are little protein building blocks, just first of all linked together and then folded intricately into a functional shape. All right, so the importance of enzymes in living systems. That's maybe too much zoom. I hope that is a sufficient amount of zoom for everyone to see. All right, great. Okay, so what are enzymes doing for biological systems? Why do we need them? So the only way, so I saw someone wants me to give permission to everyone to record this session. That's just going to eat up more time right now, and I really just want to get through this review session. So you're going to get access to the recording later. It's going to be posted and sent out, so that will have to suffice. So let's move on with the review. So what is the importance of enzymes in living systems? Right, so it's to speed up chemical reactions, because otherwise they would happen eventually, but it would just be at such a slow pace that life wouldn't be able to exist in the same way that we know it. So enzymes, they speed up chemical reactions. They just help things occur more efficiently and with a more like consistent, consistent regularity. So if you had a high fever for a period of time, do you think that that would alter your cellular function? And make sure you can explain why you think it would or would not. So we know that heat denatures enzymes, which leads them to stop working. So how would having a high fever for a long time affect your cells? So let's try to put this into words. How would a high fever affect the function of our cells? Okay, yeah. So, anything that enzymes do would be impaired. So, the enzymes in our bodies do all kinds of things. One of them would be break down proteins or build up proteins. Really, any chemical reactions that occur in the body would be impaired if there was an increased temperature within the body. So we could say that high fever would impair the body's ability to carry out chemical reactions of all kinds. And if these are chemical reactions that help keep us alive, you can imagine that very high fever for a long period of time could potentially really harm you and perhaps even kill you. All right, I see some more people have joined the session. So if you've not already, please follow this link to record your attendance. You only need to do this once. I'm going to keep posting it because I know people are going to be kind of filtering in and out of the session. But once you filled out that form for this session, you don't need to do it again. This is more for people who are coming in a little bit later.
Thanks.
All right, so enzymes have specificity. And what is it that determines that specificity? Okay, yeah, structure. So the, is it the structure of the whole enzyme or, oh, I see Diego has it. Yeah, structure and shape of the active site. Yeah. The active site, that's the kind of business end of the molecule. That's what's really going to be engaging in the chemical reaction and allowing it to occur. So we really don't care what happens with the rest of the enzyme structure. It's about that active site. That's what gives it the specificity. Okay, and here we have a multiple choice answer because if you're on the PDF like fillable form. So enzymes increase the rate of reaction by, is it by lowering activation energy, by increasing activation energy, by providing activation energy, providing ATP, or by generating heat to speed up molecular movement? So which one of these is the correct answer? Yes, it's going to be by lowering activation energy. So you're kind of lowering that threshold that needs to be achieved in order for this chemical reaction to occur. And by doing so, enzymes can increase the number of reactions that are occurring in a given amount of time.
Professor, can I just ask a quick question?
Yeah.
I thought you just said if you're looking at the form online, are we able to look at this worksheet online?
Yeah, so it should have been posted or like provided to you by your instructor. Although some people have already informed me that they did not get it from their instructor.
Yeah, I don't think we did. Okay.
So I sent it out to a few people at the beginning of this session.
I'm sorry, I came in a few minutes late. It's fine if I can't get it right now. I just was hoping it was available somewhere.
Okay, yeah. So yeah, your instructor should have posted it on like Blackboard or something. And so I don't know what to do if they didn't do that. That's
okay.
I need to wag my finger at one of my colleagues, I guess.
If I email her, she should have access to it.
Oh yeah, absolutely. Yep.
Okay. Okay, thank you.
Mm-hmm. Okay, so we've been talking about heating enzymes in question 2. So in question 5, we're talking about going in the opposite direction and cooling down an enzyme. So if you keep it at 0 degrees Celsius for an extended period of time, and then you bring it back up to whatever its optimum temperature is, what do we expect of that enzyme's activity? Really what this question is asking is, does cold temperature denature an enzyme?
No.
Yes.
No, yeah.
No.
So, yeah, so cold temperatures do not, well, cold temperatures do not affect the structure of an enzyme. Therefore, if you cooled it down and then you brought it back up to its proper temperature, it should still work. because the structure hasn't been altered at all by cold temperature. This is why you're able to like freeze an enzyme and then take it out to use in the lab as needed. Bringing it back to optimal temperature would restore its function. All right, so what are 3 factors that can denature an enzyme? We've already mentioned temperature.
pH,
Yes, pH.
substrate and enzyme concentration.
And yeah, so well, substrate and enzyme concentration, that's not going to denature a protein, right? So by denature, we mean like unfold its structure, which will invariably affect its function. But what's another like environmental condition that can affect its structure? Yeah, that's going to be salinity, or also say salt concentration. Very good. All right, I'm going to scroll down a little bit. I'll try not to go too fast so you all can still see some of the past answers. All right, how would we define the optimum pH of an enzyme? Because it's not going to be the same value for every enzyme. So how would we create a definition for that concept? Okay, yeah, Hawani, that's a very good definition. So optimum pH, pH is the pH at which the enzyme activity is highest, or yeah, most active. Or the pH at which it works the best. Yeah, so for some enzymes, like even in our own bodies, we have some enzymes whose optimum pH is 2. Those are enzymes in our stomach. They work in a very acidic environment. And then we've got other enzymes that like to be a little more neutral, and their optimum pH would be 6 or 7. So it's going to vary depending on exactly what that enzyme has to do. Why is the pH of the environment important? Well, we kind of already inferred what that reason is up here with this question. So a lot of these questions, their answers kind of overlap a little bit, but it's really just to give you an opportunity to practice kind of like phrasing these things in different ways to make sure you understand the concepts. So pH of an environment is important because why?
If pH is too high or too low, an enzyme can denature. Yeah,
exactly. That's a perfect way to put it. Yeah, if high or too low, the enzyme will denature. And when you lose that structure, you lose that function. Do all enzymes have the same optimal pH? No, they don't. Very good. No, it depends on, what the enzyme's function is and where it's found. All right, I see the number of participants has changed again. So once again, I'm going to post the link to record your attendance. You only have to fill that out once, but make sure you fill it out so that you can get credit for being here. All right, let's move to question 8 here. So we don't need to describe the steps, I don't think, but let's just for the sake of reminding ourselves, what are the steps of the scientific method? So we always start with a question, and then what comes after a question? Yes, Elijah, you are correct. Yeah, so after a question, we use that to formulate a hypothesis. And then yes, from our hypothesis. So sometimes you can fit like prediction in here too, because our hypothesis, based on our hypothesis, we're going to make a prediction of what we, intend to see in our experiment. So we do our experiment, we collect some data. And then we use that data to make a graph or a table, which we call, results. And then our final summation of all this is a conclusion, writing a conclusion. Observation. Sometimes people put question first, sometimes people put observation first. I find that these two activities are kind of, two sides of the same coin, that observing something leads you to question it, or your questions are based on observation. So you can always put observation as the first activity. Do we need to explain or expand on any of these? Are any of these steps unclear to anyone? Speak up now, otherwise we'll just move on to the next question. Although if you did this amylase activity in that lab report, then you're probably totally fine with the scientific method and how to use it and what it all means. All right, so I'm going to keep going. All right, so this graph and table always kind of annoyed me as far as this review packet goes. But basically, okay, so there's a graph showing us amylase activity according to temperature and how it changes at different temperatures. And then it gives us this table, and it wants us to use some of the words from this word bank to fill out the table. You don't have to use all of these words. But okay, let's try to do this together. So at 4 degrees Celsius, we're talking about this first data point here. First of all, let's talk about the function. What's the activity of this enzyme here at this cold temperature?
Low.
Yeah, it's low, almost inactive. We can say low or inactive. Right. What about its structure? What can we assume about its structure at that low temperature?
That denaturing maybe?
So it does not denature. So cold temperatures do not denature enzymes.
Just only high, okay.
Yes, yeah. So using some of these words maybe to prompt us, we could say at this temperature, it's still going to be properly folded. It's just not going to have any activity. So it's sort of dormant. We can add that here. So it's not active yet. It could be. So as you heat up this enzyme or heat up the environment in which you find this enzyme, we see the activity increases up to a certain point around 25 degrees. And then it starts to decrease as we keep increasing the temperature. So at 85 degrees, which is our last data point, now we can really say it's inactive because its activity is completely dropped to 0. So it's definitely inactive here. What is its structure like at that temperature? Why has it lost its activity?
It's denatured.
Yeah, exactly. It's denatured, so it's completely unraveled. It has no none of that specific structure that it needs to carry out its function. Right, yeah, so it's denatured. You can also say unfolded. Right, the chemical bonds that were holding it together are now broken. And say no more 3D structure. Exactly, yeah, so that's basically the point of this table here, is just to get you to make those connections between the temperature, the structure, and the function of an enzyme. All right, so we're moving on to page 3. So here we get to do a little bit of calculating. Oh, no, maybe not yet. So we get some numbers. Oh, but this is about the DNS assay. That's right. Okay. So if we had 1 milliliters of that fungal amylase enzyme and added it to a starch solution, and then we keep it at 45 degrees for 15 minutes. So we're giving time for this fungal amylase to break down the starch. Now, if we were to do a Benedict's assay on this mixture, what would the result be? Would we get a positive or a negative Benedict's result?
It should be positive.
Yeah, why would it be positive?
Maltose is a reducing sugar.
Yes, exactly. Right, so remember that the Benedict's assay, that's that color change assay that you did in the very beginning of the semester, and it reacts with reducing sugars. So if you let the amylase enzyme break down starch, you're going to produce smaller sugars that are reducing sugar. So it would react, it would give you a positive result with this Benedict's assay. So would, referring to the same mixture, would it produce a color change if we heated it with DNS? So we already know that there's maltose in there.
Yeah, the color will change. It'll get darker.
Right, yeah, so yes, we would get a color change because the maltose reduces the DNS, right? So when you did that enzyme lab and you're studying the, the fungal, bacterial, and human amylase enzymes, why did you use a DNS assay instead of the Benedict's assay? If they both give you a positive result, what's the benefit of one over the other?
Benedict's assay is qualitative, but the other one is quantitative.
Yeah, exactly. Benedict's assay is qualitative or qualitative. So it just tells you if maltose is present. It doesn't tell you how much is there. So that's the benefit of the DNS assay, is that it gives us a quantifiable result. Quantitative. So it tells us how much is present. So that is the real asset of that particular assay, is that it allows us to quantify the amount of maltose produced. So I can write that out as well. DNS assay allows us to quantify because it does not want me to. Oh, there we go. Okay, so what if we decided to perform a Biuret assay on the enzymatic solution? Why would it test positive?
Amylase is a protein.
Yeah, exactly. So we do still have a protein in this mixture. It's not just maltose or starch. So because it's a protein, it contains peptide bonds, which will react with that Biuret assay reagent. All right, so let's do some unit conversions. So 350 microliters is how many milliliters? So we're going from micro to milli. Milli is a larger unit. Yeah, so you're basically moving the decimal place over 3 places. So the answer here is going to be 0.035 milliliters. Okay, and then I guess it's asking how many milliliters are in 500 microliters.
.5.
Right, yeah, 0.5 milliliters in 500 microliters. So it's always good to brush up on those conversions, especially if you're not as familiar with those. All right, now we're going to get into some fun stuff with some calculating. So this is something you can definitely expect to see on your exam, where it'll give you a data table that's incomplete, along with some information about, how these solutions were made, and then you will have to calculate concentration of maltose or something else, something similar. All right, so it tells us that we have a volume of standard maltose, 5 milligrams per milliliter, and that this stock solution was used to make the solutions in column 2. The final volume of solution per tube was 10 milliliters. And so now we need to figure out the concentration of maltose here. We're going to do that using the C1V1, C2V2 formula. So these formulas will not be provided on the exam. These are things you'll have to memorize. So just a little heads up, you might want to write that down for yourself. Okay, so I'm going to scroll down a little bit. Let's create a little text box. So let's set up this calculation. So we've got C1V1 equals C2V2. So our initial concentration is this 5 milligrams per milliliter. So 5. I don't think this is the only formula. I think there's a few more. I'm trying to think. I probably have a note for myself somewhere of the formulas that you need, and I can look that up and provide it for you just to help you out a little bit. But maybe we'll wait until we take a break to do that. Right, V1 is our unknown. All right, so then C2 is going to be whatever's in this column, right? This is our concentration that we are trying to make. And our V2 is going to be the 10 milliliters. So let's start with the 0.2. So 0.2 milligrams per milliliter times 10 milliliters. All right, so this is basically what you're setting up. So you're solving for V1, and each row of this table, you're going to use these values and plug them in here. So what is the solution for this particular setup? V1 is going to equal 0.2 times 10 divided by 5. Oh, I think I set this up incorrectly. I'm so sorry. Okay, so
Professor,
yeah.
yeah, I think the concentration of the metals is C2.
Yes. Yeah, so we should be solving for, yeah, I'm sorry. I confused this with concentration. I'm so sorry about that, everybody. C2. We have V1. This is our V1. That's the amount of maltose solution that we're using. And this is the concentration of that solution that we're using. We want the concentration mgs per milliliter in this column. Thank you. I was beginning to wonder why my answers were not aligning with what I wrote here. Okay, so the 0.2 milliliters is over here. That's why milliliters are going to cancel out. It provides us with our mgs per milliliter. I'm so sorry about that, folks. All right, let's try this again. So C2 is going to equal 5 milligrams per milliliter times 0.2 milliliters divided by 10 milliliters. So we are trying to get a concentration because this is our table, this is our missing column in the table, and it tells us the units are mgs per milliliter. So we know that that's a concentration, not a volume. Sorry about that.
So we're looking for C2.
Correct.
Okay.
Yes. Divided by 10. Okay. Yes, so now C2 should equal 0.1 milligrams per milliliter. So that is the concentration of this tube here. And yes, so if you wanted to do the next row, you would just replace the V1. So instead of 0.2 milliliters, the next row would be 0.4 milliliters. So we'll put that over here. So let's say it's 0.4. So 5 times 0.4 divided by 10. What is the solution there going to be? Yeah, Camille, you are correct. That would be 0.2 milligrams per milliliter. All right, so this, if we complete this table, so the next one would be 5 milligrams per milliliter times 0.6 milliliters divided by 10. Yes, it's going to be 0.3. And yeah, exactly. Next one's going to be 0.4. Very good. Yeah, and this first one where it tells us that we don't add any of that maltose solution to our mixture, the concentration there is going to be 0, right? So this is definitely something you can expect to do on the exam. So memorize this formula and also how to use it. And you can always kind of double check your calculation by looking at the units and making sure your units align with what you're trying to solve for. That's sort of how I caught my mistake just now, is that I realized we're trying to get our answer in milligrams per milliliter. Okay, that's a concentration, not a volume. All right, and so now it asks you to graph this, I believe. Yes. So it wants you to use this information to make a maltose standard curve. So if we're graphing this, so these are going to be our 2 axes, right? Concentration of maltose and absorbance. So which of these is going to be the x-axis, which is going to be the y-axis?
Concentration of maltose is the y-axis.
All right, I'm going to have to disagree with you on that one. So we have to think of, which of these variables is dependent on the other one? Is concentration dependent on absorbance, or is absorbance going to be dependent on concentration?
Absorbance on concentration.
Right, yeah, exactly. So absorbance, this is going to be our y-axis, and the concentration is our x-axis. Yeah, so if we go down to our graph paper. So this would be concentration maltose. Don't forget those units. This would be absorbance. Which doesn't have any units. So you can just say absorb, or you can say absorbance at whatever the wavelength is. Is it 540 nanometers or something? And you already know that when you make a graph, especially if you're making it by hand, then you want to, make your intervals between your numbers regular so that your data points make sense. So basically, if you were to graph this out by hand, you should get something. You should get, the points should line up roughly in a straight line where as you increase the concentration, the absorbance is going to increase as well. So I'm not going to go through all the trouble of making a perfect graph. I'm just kind of making an example of what it would look like if we graph this. So you'd have your points and then you can draw your line of best fit. Ooh. Something like that without the little hook at the end. So, okay, once you make this, it would want you to figure out your line of best fit. And this is something that on the exam, it will give you, especially if you're doing it by hand. I don't think they expect you to get the line of best fit or like calculate the equation of the line by yourself. I was looking at the exam the other day and I believe it gives you the equation of the line, but then you have to know how to use that to get the concentration of an unknown sample. So let's say we give you the equation of the line. Oops. Yes, I can repeat that. Okay, so let's say, this data was graphed and I calculated the equation of the line. And here's what the equation of the line is. It's going to be y equals 2.9x plus 0.00002. There's a whole bunch of zeros in this. Okay, so if this is the equation of the line, and then I give you an absorbance value, can you calculate the concentration of maltose for an unknown sample? So we have this known value. We're trying to calculate this unknown value. So you basically have to know how to take this absorbance and plug it into the formula. So this absorbance, we already established the absorbance is on the y-axis, right? So this absorbance value is going to go into the position of the y, and then you're going to solve for x. And when you solve for x, that will tell you the concentration of maltose in your unknown sample. And yes,
I'm sorry. Can you say again where did you get the 2.9 again from?
So this is I've already like done this calculation before, like previously, that when you graph these points and you calculate the line of best fit, that's where this equation comes from. So I'm not going to make you go through the trouble of calculating it yourself. And I believe on the exam as well, you don't have to create this formula. It's provided to you, but then you have to know how to use this formula to find the concentration of an unknown sample. Yeah, so this is not like an arbitrary formula. It's one that I have calculated previously from these data points. And yes, the people in the chat, Javier and you are correct that you are going to use the 0.7, your absorbance of the unknown sample, and plug in for Y, and then you solve for X, and that gives you 0.24 milligrams per milliliter. So that is the concentration of this unknown sample based on your standard curve data.
Do you need to know how to get the maltose solution or no?
No, because
Okay.
that's like, well, yeah, so the unknown sample assumes that we added like the amylase enzyme to a starch solution.
Right.
Yeah, like our maltose solutions, these are like a standard against which we are measuring the activity of this enzyme. So yeah, this
Okay.
stays blank.
Okay, that's what I thought.
All right, and then while we're letting that sink in, if you've arrived late and oops, you have not signed into the session, I'm going to post the link again so you can record your attendance. You only have to do that once. So if you've already done it, don't worry about it. It's just for folks who have shown up since the last time I posted it. I want to show you something really quick with these absorbance values, and just when you're calculating the concentration for this unknown sample. So if you look at this absorbance value 0.7, and then the absorbances from our standard curve, so we know 0.7 is going to lie between 0.6 and 0.8, right? That absorbance value is going to be between these two absorbance values. So that tells me that the concentration associated with 0.7 absorbance, that concentration is going to be kind of halfway between these two concentrations. I would guess that the concentration would be 0.25 milligrams per milliliter because it's halfway between these two. Okay, 0.24, that's really close. So I know that my answer is in kind of the right neighborhood of answers. All right, so we drew our standard curve, and then we were able to find the concentration of maltose for that unknown sample in our table.
I have a question.
Yeah.
After you found the concentration of maltose for the unknown sample, would you go back and put that on the graph?
If it asks you to on the exam, then yeah, I would put that on the graph. So I know I didn't provide any numbers here, but you know, if I had numbered my axes, then yeah, I would find 0.7 over here, you know, maybe make like a dotted line or make it a different color to show like where it falls on the concentration. Yeah.
Okay, thank you.
All right, so that is it for the concentration stuff. So Elijah, not exactly. So standard curve, I guess the line of best fit kind of shows us the standard curve. I don't know, the concept of a standard curve is just using a standard set of measurements, right? Like we measured exactly how much maltose is in these solutions. And then we want to make a graph, which turns out to be a best fit line. but that graph is our reference for finding the concentration for unknown samples. So I wouldn't say that they're like synonymous terms, but they are related concepts. That we made this line of best fit using standard curve data points. You're welcome. Yeah, I hope that was illuminating. So Daniel, your teacher should have provided you this review packet, either in person or posting it on Blackboard. So I'm coming to realize that a lot of you do not have this, which is a shame, because that means your teacher kind of dropped the ball, or I don't know what happened. But if you have a Blackboard course page that there's content on, check there like first. So if you have a folder of like lab material or something, check there and see if this review packet is there.
Was this provided for the first exam?
Yes.
For the first lab exam? Okay, so I didn't get one for then either, and I already checked Blackboard. I emailed both my professors, but do you, yeah, I didn't see anything. We didn't get anything like this.
Oh my goodness. Okay. Yeah, that's a major issue then, because I'm going to let the course coordinator know that this happened. Just out of curiosity, who's your professor for 150?
For lecture, I have Professor Alam, and then for ALAM, and for the lab, I have Professor Hu, HU.
Okay. Yeah, so I don't know, I will inquire about that. And I don't know what to do in the meantime, because I don't want to like stop everything to send out.
No, of course not.
Review
Yeah.
packet. So yeah, I would say just, yeah, for those of you who don't have it, yeah, reach out to your professor and say like, hey, you didn't post this, I need this.
Yeah.
Yeah, we'll just move along.
Okay.
Yeah, because this session's being recorded, and I'm going to convert the recording into like a little YouTube video. And then once I have the link, I will send it out to the 150 professors, and they'll hopefully get it to you. And okay, so moving on. So this next part of the review has to do with that bioinformatics exercise. So if you are in an in-person lab class and you're going to be taking your exam in the classroom, there'll be one section where you'll be called up to a laptop that's set up for you, during the exam time. So you'll be called up one by one, and then on your exam will be instructions of like, go to the NCBI website, enter in this code, and then you have to answer a couple of questions about whatever protein that is. So it might ask you how many residues, or like how many amino acids long is that protein, or what organism was that protein derived from, or what are the first few amino acids in the sequence. So you'll have to answer a few questions about this unknown protein using the information you find on this website. Yes, you'll go onto a computer to do that. So that will be like one of the like stations that you go to during the exam. So the NCBI site, you should know that when you enter in one of these little codes, which will be given to you on the exam, you have to enter in the code, make sure you are searching a protein database, and then it'll spit some results out at you, and you just need to know how to kind of interpret what the screen says. So, okay, this particular protein has 499 amino acids. It's from the Aspergillus oryzae organism. And if I was asked, what are the first 3 amino acids in the sequence, I would have to click on FASTA, and that would give me the 1-letter amino acid code for the entire protein. So how about we do this together right now? So everybody, open up a separate tab. And you can just Google search NCBI and the site should come up, but use this code, use this accession code, and then tell me about what enzyme this is or what protein this is. All right, so it looks like there's 41111
Okay, good.
248A. All right, so make sure you're searching the protein database. 248A. All right, so if you went to the NCBI site and entered this in, yes, very good, Melanie. So this protein, it has 512 amino acids in its sequence. And what organism is it from? So we are entering in this number here. So it's 4 ones, 1, 1, 1, 1, and then 248 and the capital letter A. So if you go to the NCBI site, which is pictured here, make sure you've selected the protein database and input that number here. And yes, very good. It's from Bacillus licheniformis. It's an, alpha amylase, heat stable. Awesome. Okay, what are the first 3 amino acids in the sequence? You can just give me the single letter codes. Very good, Melanie. Yeah, it's going to be MKQ. Yeah, so if you were able to do that, then you're going to be fine on the exam. All right, so it asks us how to obtain the FASTA sequence. Yeah, sure. And I'm going to write it out here too. So obtaining a FASTA sequence. So first of all, you go to the NCBI homepage. Select the protein database. Enter the accession number, which is kind of like the catalog number for a protein. Click FASTA, and that's basically it. So what I can do right now actually is I can switch over to the NCBI website just to show you directly what we're doing. So let me do that. Okay, I'm going to stop this share just for a moment. And I just want to share my browser window. Come on, man. Here we go. Okay. So you should be seeing my Google browser. And you can literally just search NCBI, and it's the first thing that comes up, the National Center for Biotechnology Information. So this is the NCBI homepage. So if you're here, you're going to, first of all, click here where it has all databases, and you want to look for the protein. All right, so you select protein, and then this is the search bar where you enter the number. 8A. Then you hit search. And then it spits out the information. So this is an alpha amylase that's heat stable. If you go down here, it says source organism. So it tells you exactly what organism it's from. Up here, it tells you the number of amino acids in this entire protein. And then if you want to actually see that sequence of amino acids, then you click this link here that says FASTA. So when you follow this link, it takes you to, should open up, yeah, there we go. And so this is the entire, these are all 512 amino acids. Each amino acid is being represented by a single letter. And so each letter tells you which amino acid is there. So one of the questions you might see is, what are the first 3 amino acids in this protein sequence? And so then you would say MKQ. All right, so that is how you use the NCBI site. Now, the other site you might be asked to use is the PepStats. So actually, that's on the next page of the review packet. And yeah, let's. So the Emboss PepStats website looks like this. And so this is where you could enter in that amino acid sequence. So there's a readout already for you on page 7 of this review packet. So I'm just going to switch over to that. So let's switch over to the packet. There we go. Okay, so we were up here, we did this thing, and now the PepStats is basically analyzing this sequence of amino acids. And so they've done this for you here. So if there's a readout like this, where it analyzes that amino acid sequence, you're supposed to know kind of how to interpret this information to answer questions. So this just tells you like how many of each amino acid is in the sequence and how many of those amino acids are polar or non-polar or acidic or basic. So that is what it's asking here on this table. So for this Emboss PepStats information provided on page 7, how many amino acids are there total? How many are polar? How many are non-polar? So if I just leave this up here, and that is kind of what is highlighted in these boxes. So residues, that's just another name or like another term for amino acids. So when it says residues equals 478, that's telling you the total number of amino acids in this particular protein. And then down here, so it gives us the number of non-polar and polar amino acids, as well as the relative percentage of non-polar to polar amino acids. And Camilla, to answer your question, yes. So if you were on, the NBOS PEP stats, in order to get this readout that we're looking at here, you would copy basically the FASTA sequence. You would copy that from the one site into the search box for this site. And then it like instantaneously analyzes that amino acid sequence. It gives you the stats of that peptide, which is why this site is called PepStats. Yeah, so we have 478 amino acids. (212) are polar. 66 are nonpolar. Oh, here we go. And then got 44% and about 56%. Yeah, exactly. Yeah, so this is just about, making sure that you know just how to kind of read this bioinformatics readout, right? So it spits out all this information, but can you sort of read this information well enough to complete a table or just to answer simple questions about that protein?
Is that 55% or 56? I think it's 55.
Oh yeah, it says like 55.6. I was rounding up, but yes, technically it's 55.6, and then this one was like 44.3 or something. But yes. All right. So moving along. Okay, so here it's shown you some data that's been collected on 3 species of amylases, so the human, fungal, and bacterial. And it basically, so here, it doesn't really put it in a question format, but it's asking you to analyze the data and explain how amino acids outside the active site contribute to the function of the enzyme. So we see that the 3 amino acids within the active site are conserved across all 3 species. So the active site is identical, but these proteins still differ quite a bit, right? So some are larger, some are smaller, and they all have like a different optimal temperature and pH. So basically, how is that possible? How, what is the role of amino acids that are not in the active site, basically? Oh, to here I use, so for the PEP stats, the percentages are at the kind of at the bottom of the screen or at the bottom of this chart. So I'm going to scroll up a little bit. So this is the PEP stats readout that's provided. So if you just, you input a sequence of amino acids and then it'll give you this data about it. So it'll tell you the total number of amino acids, and you have to scroll to the bottom for it to give you this little chart where it gives you the property and then how many amino acids have that property and then the percentage. So that would be if you go to PepStats and then put in a sequence of amino acids and hit search, then it would give you this readout. I guess. So the PEP stats, I feel like the PEP stats, they might just give you a readout. I'm trying to remember what the exam said. You definitely have to go visit the NCBI site. I don't know if they make you visit the PepStat site as well, just because that's a little bit more involved to like put in the sequence of amino acids. Because when you did the bioinformatics exercise, that was the one where you had to like, remember, like copy and paste those amino acid sequences onto like a separate document. And then you would like copy them from that document into these various websites. So I don't think you have to do that on the exam. I think you only have to visit the NCBI. But they want you to know how to like read the results page basically of the PepStats site.
Oh, okay.
Yeah, I hope that, that clarifies things. Yes, the recording will be provided, and thank you for letting me know that you are late. I can post the attendance link. Whoop, that's not the attendance link.
Excuse me, Professor.
Mhm.
I think I didn't sign in the attendance sheet.
Yes, I just posted the sign-in link.
Okay, thank you.
Mhm. All right, so let's go back to this question here. What is the role of amino acids not in the active site? We know that the amino acids in the active site are hugely important, which is indicated by the fact that they are conserved across 3 very different types of organisms. But what about all the other amino acids? You know, what's the point of having like 680 amino acids that are not part of the active site? All right, what do we think? Do we have any educated guesses as to what the purpose of these other amino acids are? The ones not in the active site? All right, so I'm feeling like maybe we're a little stumped here. That's okay, or maybe we're just afraid to share our responses. That's all right too. So amino acids that are not part of the active site, their job is to just stabilize the rest of the structure, especially when you have enzymes that need to function at extreme temperatures or extreme pHs. You want you want the overall structure to remain intact despite those environmental conditions. So all these other, hundreds of amino acids that are not part of the active site and are not actually like catalyzing chemical reactions, they're still important because they stabilize. Whoop, I need to do that. Stabilize the shape of the enzyme in different environmental conditions. All right, so just because they're not part of the active site doesn't mean they're totally useless. They are still there for a purpose, and it's usually just to help maintain the overall shape of things despite whatever pH or temperature it needs to survive in. All right, so now we are moving on to exercise 10. So this was PCR and restriction digest, electrophoresis, all that fun stuff. So what are the main steps of a PCR reaction? There's 3 main steps. And what is happening in each one?
Denaturation, annealing, and extension.
Great, yeah, excellent. Denaturation. Then we have the annealing and then extension. Okay, so denaturation, what is being denatured here in this first step?
Your DNA strands separate.
Right. So yeah, the DNA double strands separate. And what is breaking those hydrogen bonds between the DNA strands?
High temperature.
Yeah, exactly. So the double strands separate due to high temperature, which is what, like 95 degrees. Okay, so then the annealing step. What is happening in the annealing step?
Your primers bind to the target sequence.
Beautiful. Yeah, primers bind to your target sequences. This is happening between 50 and 60 degrees. And then extension.
The enzyme Taq polymerase extend primers by adding NTS to primers and copying the strand.
Yeah, absolutely. So yeah, Taq polymerase extends the primers, copy the target sequence. Oh yes, and using the DNTPs, so like the free nucleotides that are in the solution. And this is occurring at about 72 degrees. Kumar, to answer your question, we did not skip 8 and 9. I think they are later in this file. It's going by, I think, like the order of doing the exercises, and usually you start exercise 10, then you do like 8 and 9, and then you follow up with 10. So I think that's why they put it in this order. Although I agree, it's a little bit confusing. But no, we didn't skip 8 and 9. We will still go over those. Let me see, I'm just going to zoom out really quick. Got 10, 10, 10, 10, 10. More 10. Oh, there we go. And then 9 and then 8. Yeah. So for some reason, going a little backwards. All right. Yeah, so we did not skip them. They are still coming. Whoa. All right. Okay, those of us just joining us, make sure you sign in. I'm going to post the link again. You only need, oops, you only need to sign in once. Oh, goodness, it's so annoying. Why is this? Okay, here we go. All right, why does this Taq DNA polymerase enzyme not denature at these high temperatures?
Because it's thermostable.
Yes. So it's a thermostable enzyme. And because it is coming from an organism that naturally lives in a very high temperature.
Yeah, would it be okay if we said that, like on the test, that it comes from, you know, originates at a very high temperature, like it originates from hot springs?
Yeah, absolutely. Yeah.
Okay.
So yeah, you can say it comes from an organism. Yeah, that lives in hot springs, or you can say lives in a hot environment.
Mhm.
Yeah, any kind of iteration of that statement is totally fine. And if you want to, if you're up on your Latin spellings and you want to name the organism and say it's from Thermus aquaticus, you can do that too. Aquaticus, yeah. But yeah, that's why you can heat it up and it doesn't denature, and then you can keep heating it up and cooling it down over and over again, and it just keeps working. So definitely beneficial there. And I don't know if you have followed these links yet, but these simulations here of like PCR and restriction enzyme digest and electrophoresis, these are really great kind of like interactive things to like click through. So I guess it would help if you had this handout, but we'll solve that problem. We'll figure that out. But in any case, I think you could probably just search like lab exchange and then these keywords, and you could probably find it online yourself. But they're really great, yeah, like depictions, animations of like what is actually going on at the molecular level. So I find that it really helps kind of visualize what these events are that are occurring. All right, so what are the main differences between regular DNA replication and steps of PCR? So we can do like just a little comparison. So we'll just say DNA replication. We can name some things about DNA replication and how it's different from PCR reaction.
Professor, for the DNA replication, is like they take the whole chromosome, and for the PCR version, they just take like a small piece or like a sequence.
Yeah, excellent. That's one big difference between the two. So yeah, in DNA replication, we're copying the entire genome, whereas in PCR, we're only only copying a small segment or a target sequence. And then there's one more. There's one more big difference that you could name.
Is it speed?
Not speed, but the number of copies. So in DNA replication, how many times are we copying the entire genome?
2.
Well, we're copying it just once, but yeah, we're going to end up with two exact copies, right? But basically the replication process, each replication process, you're only making one copy of the genome. And then how many copies do we make in a PCR reaction?
A lot.
A lot, yeah.
Exponential.
Oh yeah, that's a good word to use. Only copying a target sequence and you're making exponential number of copies. Right, so the number of copies and what you are copying, those are two big differences. I guess you could make an argument for temperature if you're like trying to compare our cells versus PCR. That can also differ. So I would argue that those are the main differences. All right, so here is another little math question. How many DNA copies would be theoretically made if you allowed the PCR to go through 20 cycles? So there's a way to mathematically express how many copies you could make. What is it?
Is that n exponential 20?
Yes, yeah, so it is, yeah, 2 to the power of 20. Which is what, 1, 0, 4, 8, 5, 7, 6. We are on page 9. So we're at the top of page 9 for this review packet. And I know we're going through the last exercise, exercise 10. We've not gone through exercises 8 or 9. Probably go over those after taking a little break. I usually like to take a break halfway through this review session. So yeah, probably in like 20 minutes, we'll take a little like 10 or 15-minute break. Just get done what we need to get done and then come back to it. But okay, so this this mathematical expression, so if we wanted to write this out or kind of explain it, so the number of DNA copies equals 2 to the n power, where n equals the number of PCR cycles. So that's how we got 2 to the 20 power, because it says that we put it through 20 cycles.
Hey, Professor, can we write on the exam the n is greater than 1 million or whatever instead of the whole numbers? Is that possible?
I think, well, on the exam, you would want to write this exact value because since we learned how to calculate it precisely, I think it would be best to give like that precise value, not and not say like over 1 million.
Okay, thank you.
So, okay, so Kumar, so this is page 9 according to this file. So I also have an older version of, so I have this version, I think this is the more current version of this file. I have a printout of an older version of the lab review packet. And I think Kumari, you and I have this. Yeah, so, oh, then so maybe I sent you the older file. Well, all the questions are the same. It's just in a different order. So if you go more towards, yeah, if you just skip forward a little bit, you'll find these same questions. They come after the exercise 8 questions. I'm sorry for that confusion. So like on my file, I'm on page 11, and on this file, it's page 9. I really thought I sent you guys the newer file, but perhaps I did not. You are allowed to use a calculator during the exam, although it has to be like a basic, like tabletop calculator. You can't use your phone, and you can't use a graphing calculator. And they have calculators in the lab classroom, and I'm sure they'll be sanitized in between uses. So they'll have them available for doing these simple calculations. Oh, darn. Yeah, I did send you the older one. I'm sorry about that, Kumari. But like I said, the questions are the same. It's just in a slightly different order. I apologize for that.
Professor, during the break, is there any way you could email it to me?
Yes, I can do that during the break.
Thank you.
The reason you can't use graphing calculators is because you can program graphing calculators, like so you could create a program that has like the formulas that you need for this exam, and it's a way to cheat and not have the exam or the formulas memorized. So that's why they don't allow graphing calculators. Okay, so this next question asks about those 2 alleles for beta hemoglobin that we studied. So the HbA and the HbS. So what is the difference between these 2 alleles? So by alleles, we're talking about the sequence of DNA, right? So what is the difference in the sequence between HbA and HbS?
Professor, I will say like the difference is the mutation.
Right, so there is a mutation, yeah. There is a mutation in the HbS allele. And where is it exactly?
I think the seven.
Yes. So there's a mutation in the HbS allele. So we'll say there is a nucleotide substitution. Oops. Substitution in the 7th amino acid. And even more specifically, it changes a glutamate to a valine. That is the substitution that occurs. And how does this substitution cause sickle cell disease?
This causes like a whole new amino acid to take place.
Right, so if there's a different amino acid in that position, what is the consequence for that protein? What happens to that protein then in the red blood cells?
It goes through a missense mutation.
So yeah, it's a missense mutation. It causes it causes something to happen to that protein. What does the protein do? Yes. Oh, Diego, you said it. It makes the amino acid hydrophobic. Yeah. Makes the resulting protein yeah, hydrophobic and prone to clumping together. Right, because that's really the issue there with that beta hemoglobin is when it has this incorrect amino acid in that position, it has this tendency to clump together, especially in low oxygen conditions. And when it does that, it's no longer able to carry oxygen like it's supposed to. It's just clumping together, creating these like big tangly strands within the red blood cell, altering the shape of the cell. And so that's really what gives rise to the disease, is that protein, instead of carrying oxygen, it just clumps together. So when you did the lab exercise with all of these molecular biology techniques, how were you able to distinguish between these two alleles?
So by running the DNA on the gel electrophoresis, I guess.
Right, yeah, so the electrophoresis, the results there showed that one of these alleles got cut by those restriction enzymes and the other one didn't. So which one got cut? Which one of these alleles was able to be digested and cut by those restriction enzymes?
I think the HBA, the heterozygote, or I mean the wild type. I mean, is that
Yeah,
what?
exactly. So yeah, the wild type allele was able to be cut by the restriction enzyme, which created fragments that you were then able to visualize with electrophoresis. Right, so this was the way you were able to tell who had the disease allele, who had the wild type allele. Okay, so here it wants you to choose which one of these primers would have a higher annealing temperature. How would we determine which one has a higher temperature? Oh, Anel, I just saw your question. So restriction enzymes cutting foreign DNA, that's their natural purpose. So that's how that's what they were discovered to do originally, is that they were these enzymes that existed in bacterial cells, and their job was to cut foreign DNA that entered the bacterial cell before it had a chance to infect the cell. But now in this research setting, we've been able to isolate these restriction enzymes, and we can kind of get them to do things that are important to us in a research setting. So yeah, you are correct that they do cut foreign DNA. That's like their natural purpose in bacterial cells, but that's not necessarily what they do. Yeah, when we're using them in like a research way. Camille, yes, you are correct. So the high temperature, so annealing temperature depends on amount of G's and C's basically. In a sequence, because the guanine and cytosine have 3 hydrogen bonds between them instead of just 2. So it takes more heat to break bonds between C's and G's. So basically, to answer this question, what you have to do is count the number of G and C residues in each sequence, and whichever one has more G's and C's, that's going to be the higher temperature. So how many do we have here? I count 10. And the second one, I count 8. So because this one has more G's and C's, it's going to need a higher annealing temperature. And 3 H bonds. All right, let's see if we can finish up this page before we take a little break. What is the difference between sickle cell disease and sickle cell trait? So let's say, what's the difference in genotype, and also what's the difference in phenotype?
When you have sickle cell trait, the genotype would be HBA and HBS, but when you have the disease, it would be HBS, HBS. And the phenotypes for the trait, you're only a carrier, so you don't show the disease. And for the actual disease, you have the disease. That's your phenotype.
So yeah, if you are sickle cell trait, you just carry the trait, you're not really affected. Or maybe very, very mildly affected. But if you have sickle cell disease, it means you have both of those S alleles and you are affected. And then what is the inheritance pattern of the sickle cell trait or sickle cell allele?
Heterozygous versus homozygous?
Good guess. So we're thinking, so by inheritance mode, it can be either like, it can be sex-linked or it can be autosomal. It could be recessive, it could be dominant.
This one is recessive and autosomal.
Yeah, exactly. Yeah, it's autosomal and recessive. So autosomal because it's occurring on, what is it, chromosome 11? I forget exactly, but it's not on one of the sex chromosomes, so that makes it autosomal. And it is recessive because you need both of the alleles in order to be affected. If you just have one allele, you're just a carrier. All right. Now we're going back into PCR. So what are the components of the PCR reaction? There's 4 main components. And let's just try to describe the purpose of each one.
Professor, I forgot, but I think on that day we used a buffer, DNA, and nucleotide. That's all.
Okay, yeah, so definitely nucleotides.
And Taq polymers
Yes, Taq polymerase, that's kind of the secret sauce there.
and primers,
Right, primers.
template DNA,
Yeah,
I think nucleotide.
it's annoying that it won't let me do 4 rows here. Ugh, sometimes I really hate these autofill PDFs. I'm going to make my own text box. All right, yeah, so we had nucleotides, which we call the DNTPs, our Taq polymerase, primers, and target or template DNA. So the buffer is just kind of like the solution, like the liquid that everything was mixed in. And I think the buffer also has like magnesium, which is required for like annealing and extending, as well as just, you know, the correct like ionic concentrations to keep the reaction going. So the buffer is not really considered a main component of the reaction. It's just sort of like the milieu in which the reaction takes place. So DNTPs, these are our free nucleotides that we need to create these new strands, right? We're copying strands of DNA or like little sequences of DNA. So we need the raw materials for that. We need the building blocks to make those new strands. So we already talked about our Taq polymerase, how that is needed for actually extending the primers to copy our target sequence. What do the primers do?
I think they have like forward and reverse and I don't know how to explain the
So so there's forward and reverse primers, and they basically kind of flank the target sequence. So the primers are what specify the sequence to be copied. So rather than making a copy of the entire genome or the entire strand of DNA, you're only going to copy the sequence that you're really interested in. The DNTPs, that's just free nucleotides. So that means those are just the adenines, thymines, guanines, cytosines. It's just the letters. It's just like an alphabet soup for making your new strands. That's what DNTPs refers to. And they usually already come in like a buffer solution. So like when you're mixing up your reaction, you add like your DNTP buffer, which is like buffer solution with all the free nucleotides that you need. Our template DNA, that's just going to be our DNA sample to be amplified. So we start with a sample of just whole genome DNA. Then we have our primers, which are going to specify which part of that genome we actually want to make lots of copies of. And in order to make those copies, we need an enzyme to catalyze that reaction, which is Taq polymerase. And then that enzyme, it needs something to, connect to make those new strands. It needs nucleotides. So it needs a whole bunch of adenines, thymines, guanines, cytosines, so that it can create copies of that target sequence over and over and over again. So what are some applications for PCR? So we used it during our lab as a way to diagnose these fictitious individuals, but what else could we use it for?
Cremisence.
Did you say forensics? Criminal.
Scene.
Oh, crime scene.
Yeah.
That's what you meant. Okay, sorry. Yeah, crime scene. Yeah, so for forensics, if you want to collect some kind of biological residue at a crime scene, you could use PCR to amplify that DNA. You can use it to determine paternity. Yeah, identifying unknown persons. Yep. It can be used in archaeology or in research, like if you're doing evolutionary research. So all kinds of things. All right, so we are at the bottom of this page and it is 7 o'clock. So let's go ahead and take a break. I'm going to just leave a little note here on the screen. Oop. Okay. Okay, so let's say we will restart at, let's say we'll cut the difference. We'll say 12 minutes instead of 15 minutes. So we'll restart at 7:12 p.m. So until then, you can go take a break, use the restroom, get a drink of water, whatever you need to do. So we'll restart in 12 minutes. And in the meantime, if you want to just send me your email really quick in the chat, and then I can send you the this review.
Thank you so much.
Okay, welcome back, folks. Sorry I'm a little late. So we had a question in the chat about formulas that you will need. And it's actually not that many. I feel like there are a lot more formulas you need to know for the first lab exam. The second one, not so much. So really, it's going to be that C1V1, C2V2. So calculating, the dilution concentration. You'll want to know how to calculate amylase activity. So if you're given, data from an experiment, can you calculate the rate of enzyme activity? So remember that is what is that, milligrams per milliliter per minute. So basically taking the concentration of maltose created and then dividing it by whatever that length of time is for the experiment, which I think is like 10 minutes. So it's basically taking maltose concentration divided by 10. So this will be for concentration, and then also being aware of how to use the formula for a line to get maltose concentration for a given absorbance. Accumulating. And that's basically, I think those are the only calculations you need to do. Because otherwise you're just, I'm sorry.
Did you said for the amylase activity, it's just maltose concentrations versus the time? That's it?
Yeah, exactly. Yeah, that's the concentration divided by time.
Got it. Thank
It's
you.
reflected by that unit. We did do the amylase activity on the review. Yeah, so that's what we did. Basically started out with that. So right now we're finishing up exercise 10 questions, and then we're doing exercise 9 and then exercise 8. And before I dive back into answering these questions, I'll send the packet to you, Tahira, before I get distracted. Right. Okay. All right, so this next question says, yeah, I'll leave that up here for a moment. What are nucleases and what are they made of? So that ACE ending is sort of a clue there.
The enzyme and they're made up of amino acid.
Yep, yeah, exactly. So they're enzymes. So they're made of amino acids. What else were you gonna say? Or if that was it, that's fine too. Yeah, so the ACE ending tells us it's an enzyme. The nuclea part tells us kind of what the enzyme does or what it's working on, which would be nucleic acid. So what's the difference then between an endonuclease and an exonuclease?
An nuclease cuts the sequence like in the middle, and an exonuclease cuts the sequence at the ends.
Yeah, exactly. So the difference is really about where it is doing this enzymatic reaction. So endonuclease means it is cutting within a strand. Exo means it's at the end of a strand. And by strand, of course, I'm talking about a strand of DNA, some DNA sequence. And then the restriction endonuclease, how would we define that? So we know it's an endonuclease, so it's going to be cutting within a strand, but what does this restriction part refer to?
It recognizes a specific sequence of DNA and cuts both strands.
Right, yeah, the restriction part kind of, the restriction comes in that it is only going to recognize, right, a specific sequence. It's not just going to cut anywhere or like cut in arbitrary places. So it's cutting within a strand at a specific sequence, which we usually call the recognition site. And you're right, it will cut both strands, creating either blunt ends or sticky ends. Oh, and that's the next question. Okay, so first of all, which nuclease did we use in the lab? So they all have these like little abbreviated names.
EcoRI.
Is that the one you guys used? I thought it was the D one.
It's GE.
Yeah, so DDE and then capital I, so, or one, but yeah, DDEI.
I'm sorry, I thought I was big.
Oh yeah, don't worry about it. Yeah, EcoR1 is like a very common one that's used a lot of times, like just as an example to explain restriction endonucleases. But yeah, the one that we used was the DDEI. And what kind of ends does it produce?
Sticky ends.
Right, yeah, so it produces sticky ends. And what does it mean that a DNA sequence is palindromic?
It means that when it's read backwards, like when the sequence is read backwards, it sounds the same as it's read forwards. Like when it's read from a 5' to 3', it's read the same way in a 3' to 5'.
Right, so it's the same being read. You were so close, so to being perfectly correct. You were correct up until the very end of your statement. So the sequence is the same every time it's read in the 5 to 3 direction. So it's not necessarily going to be the same if you read it 3 to 5. But if you read like the top sequence 5 to 3 and the bottom sequence 5 to 3, which is going in the opposite direction, those should be identical. So the sequence is the same when it's read in the 5 to 3 direction. And we will kind of do an example of that in this next little exercise. So it asks you to identify which of these is the palindrome. And the way you want to approach this is to basically write out what the complementary sequence is. Unless you're really good at visualizing. But I know for me, it helps to write out the complementary sequence. So this first one, AAGGTT, what is the complementary strand going to read?
It's yeah.
TUU.
No, we're still DNA, so it'll be C's, TT.
CC, AA.
Yeah, exactly. Okay, so we have both strands, and are they identical? So if we read the top one from left to right and the bottom one from right to left, is it the same sequence? No. So this first one is not a palindrome because it is different. If we read it from 5 to 3 on the top, 5 to 3 on the bottom, they're different sequences. All right, so let's try this next one. So GGGCCC, what is the complementary sequence there?
CCC, TGG.
Right, yeah, CCC, GGG. And so when we read it from left, when we read the top strand from left to right, we have 3 Gs and 3 Cs. Then when we read this complementary strand from right to left, we have the same thing, 3 Gs and 3 Cs. So that makes this a palindrome because it's read the same in each 5 to 3 direction. It's the same sequence. All right, let's do the next one. So a bunch of A's and T's. So that would be TATA.
TAT.
Right, so is this a palindrome? So AT, 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 and then the bottom row in the other direction, AT, AT, AT.
Yes.
Yeah, so this is also a palindrome. And now let's do this last one. So is this final sequence, is this a palindrome?
No.
Nope. That is correct. Yeah, so only these 2 sequences were palindromes, the other ones were not. And this is a way you can check it. And so even on the exam, you can, with your pencil or whatever, just write out the complementary sequence and see if it is the same if you read it in the 5' to 3' direction. All right, so this next question asks, how does the number of restriction sites relate to the number of fragments produced? So we can express this sort of mathematically, or kind of write an equation. So maybe we can use a visual here. So if we have a linear piece of DNA and we cut it twice, so we cut it once here and once here, how many fragments do we produce?
3.
Yeah, so we have 3 fragments for 2 cut sites. So the fragment number equals the number of cut sites plus 1, or we'll say n plus 1, and n is going to equal how many cut sites there are. All right, so let's do, if we had a circular piece of DNA, where is my cursor? Okay, so if we have a circular piece of DNA and we cut it twice, how many fragments are we going to make? So we cut it here and we cut it here. That's only going to make 1 fragment, 2 fragments. So the number of fragments is equal to the number of cut sites. So that's basically what they are getting at or alluding to in this question. So in circular DNA, the number of fragments is equal to the number of cut sites. In linear DNA, the number of fragments is the number of cut sites plus 1. All right, so let's move along. So now we are going from our restriction digest to gel electrophoresis. So what is electrophoresis? How do we define this technique?
It's the technique used to separate the fragments of DNA by allowing them to move through the gel matrix.
Yeah. So it is, oops. So it's a technique of separating fragments based on size, and we are using we're using electricity to do that, to move these molecules through some kind of, like this gel matrix. So we're using electricity or an electrical current. Maybe that's more apt, electrical current to move molecules. And it kind of answers the next question too. What does gel electrophoresis allow us to do? Well, we used it to separate DNA molecules based on their size. Let's say fragments of DNA molecules. Okay, so this agarose gel, what is it like? So if we kind of zoomed in on this molecular structure of agarose, what would it look like, or what does it resemble?
Jello.
Yeah, so it's a jello, but what creates that jello? What is agarose? I think it says in one of your, in that lab handout for that exercise. So agarose, the ose ending tells you it's a carbohydrate. So by itself, agarose is a polysaccharide, but this agarose gel, when it's formed into this gel, it kind of creates this mesh or this sieve. And that's what's going to kind of capture these different fragment sizes. We can say it's a 3D mesh. And these long polysaccharide molecules, they're going to be held together in this mesh with a certain type of chemical bond. What kind of chemical bond do you think it is? So you learned about all kinds of chemical bonds this semester, right? Learned about covalent bonds and ionic bonds and hydrogen bonds and van der Waals interactions.
Covalent bond.
I'm sorry?
Covalent bond.
I still did not understand it. The Wayland bond?
Covalent bond.
Covalent bond. Good guess, but it's actually going to be the hydrogen bonds. Yeah, so they these agarose molecules are going to be held together with hydrogen bonds, because if you heat up this agarose gel, it goes back to kind of like a liquid state because you're melting those hydrogen bonds. But as it cools, it sort of forms this jello-like material. So that gelatin is actually these molecules being held together with hydrogen bonds with enough spaces in between the molecules that these DNA fragments can slip through if they're small enough. And it's this electrical current that we're adding to the gel that helps kind of force those fragments through. So how fast things travel, we call it that migration rate. So there's two main things that are affecting the migration rate of these DNA fragments through a gel. So who can name those factors?
I only know one. I think it's the voltage.
Actually, that can affect it too. So voltage definitely does. So that's actually a third one from what I had listed here. So yeah, voltage.
Size.
Yes, the fragment size.
And the shape.
Yeah, and the gel. So the concentration of agarose you have in the gel, yeah, can also affect it. Because if there's a lot of agarose, it makes for a very stiff gel and only very, very tiny things can pass through. If you have less agarose, then it's a very loose gel and bigger molecules can pass through. So yeah, voltage, DNA fragment size, and the concentration of agarose in the gel all affect migration.
How about the shape?
Oh, the shape of the DNA. Actually, yeah, that's another one too. So whether the DNA is circular or linear, or it's what is it? There's a couple other shapes too. Circular or linear. Yeah, so agarose is originally like derived from seaweed. I think now it can be made synthetically. But chemically, it's a polysaccharide. So you're not wrong in saying it's a seaweed. But you know, seaweed is a plant and plants are made out of all kinds of polysaccharides and other biological molecules. So. All right. So when we did this gel electrophoresis, when we ran those DNA fragments through the gel, we included a DNA ladder. So what was the point of that DNA ladder?
Estimate the size of fragment.
Right, for estimating the size of the fragments. In our sample. So the DNA ladder is a standard. Right, so the ladder had like a combination of all different fragments of known sizes. So if we know the sizes of those fragments and wherever they kind of wind up in the gel, then we know kind of approximately what the sizes of our sample fragments are. So what are some applications for restriction analysis?
Professor, there is that too, like for the expression of the gene and to make insulin, and also when they want to maybe create another organism like cloning.
Right, yeah, cloning is a big one. Yeah, and for you're right about the insulin as well. Yeah. Insulin and other hormones. So along with the cloning, so just yeah, replacing genes. in a genome. So that's usually, we usually like to do that with bacteria, so we can kind of splice in genes of interest if we want to study them. And that's something that we use restriction analysis for. All right. Okay, so we're looking at these different restriction enzymes, and it's giving us their recognition site. So which one of these does not make sticky ends? So the arrows here are indicating where the enzyme cuts. And yeah, it's the SCA1 that is making a blunt cut because it's just going right through rather than through, across, and through. So all the other ones are making blunt ends, or excuse me, sticky ends. This is the only one making blunt ends. All right, pretty straightforward one. Let's go on to the next page. So it is giving us these results here. And it's saying that, okay, we got this pattern from a digestion of a linear DNA molecule by using two different restriction enzymes alone and then together. And it wants us to determine the size of the bands in two different ways. So estimating the size of the bands by just a visual comparison with the ladder, and then also measuring using this ruler here next to it. So actually, I think, yeah, on this PDF fillable one, you should be able to like type in the size right above the band. So let's see, this is our uncut DNA, right? So lane 4 is DNA only. It looks to be about 8,000. So in each lane, the size of all 3 or 2 fragments should equal 8,000, right? Yeah, that's 8,000. So what is the sizes? What are the sizes of A and B in this first lane?
5,000, 3,000, 5,000.
We got 5,000 for A and then 3,000 for B. And that equals 8,000, so we know we're doing something right. Okay, so then C and D.
$7,000 and 1,000.
Yes. 7 up here, 1 down here. All right, and then for E, F, and G.
4,000, 2,000, 1,000.
Yep. Yeah, so 4, 2, and 1.
That doesn't equal 8,000 though. Is it okay because it's close?
5, 6. Oh yeah, you're right. That's weird. Yeah, actually that's okay because I think there's a reason for that we're going to find out.
Okay.
Yeah.
Oh, Professor, I have a question. So how did we, if we have a ladder, it's easy to figure out which size, but if we don't have the ladder, then we have a ruler. It's how do we gonna measure it? It's I'm just confused with that one.
So it's basically the ladder and the ruler are like 2 ways to arrive at the same answer, or just 2 ways to arrive at like comparable answers. So for this ruler, you basically measure the distance that each band traveled, typically in centimeters. I think that's so. And I don't like this method personally because it's a lot more labor intensive. I prefer to just kind of eyeball it next to the ladder, but okay, so measuring the distance of migration of the ladder bands from the well, and then using that, kind of creating a standard curve. And then using, then you can find your distances on your standard curve and use that to like figure out what the band size is.
Yeah, because we have on the assignment is with the ruler. It's I had really hard time to figure out.
Yeah,
I know we switched it from centimeter to millimeter, but still it's yeah, it's very tough.
yeah, it's challenging. But I think it's important, not that you do it necessarily like correctly, but if you as long as you understand the like principle behind finding the fragment size this way, I think you're going to be in good shape. Okay, so we're talking about two different variables here, right? The distance from like the distance migrated and the fragment size. So distance migrated, fragment size, which one is going to be plotted on the x-axis, which is going to be plotted on the y-axis? So fragment size, distance migrated, which one is the dependent variable?
The distance is a dependent variable.
Right, so fragment size is going to be on our x-axis, distance migrated on the y-axis. And Diego, to answer your question, I would need to go back and look at the lab exam again to see if the ruler method is required on there. I want to say no, but maybe that's wishful thinking on my part. I can give you a definitive answer though at the end of our session. But okay, so fragment size along the x, distance migrated along the y. So on our graph paper here, we'd have our, no, fragment size, right? Sorry, fragment size and base pairs. And then distance migrated on the y-axis. Millimeters. And we already know kind of intuitively what the relationship between these two variables is, right? So as fragment size gets bigger, what's going to happen to the distance migrated? And yes, Nikechi, you are absolutely correct. The distance traveled is going to depend on how large that fragment is, because bigger fragments don't travel as far. Smaller fragments will go very far. So if these fragments, you know, assuming that we start down here with like smaller sized fragments, let's see, we'll do 1,000 here. Oops. One, two, three, four, 5, 7, and then 8 would be there. Okay. All right, so if we have a really small fragment, it's going to go a lot further, and a really large fragment is not going to travel very far at all. So if we were to measure it out, like take the time to measure it out, which would mean, and I need to use a piece of paper for this because I'm looking at a computer screen like we all are, right? So I would take a piece of paper against the computer screen and I would say, okay, so this 8,000 base pair of, you know, this ladder one right here, it lines up with, it looks like 11 centimeters, or sorry, 1.1 centimeters, which is 11 millimeters. So that'd be, so we're ending probably here. And then let's see where our 1000 goes. 1000 base pairs. I'm looking at 43 roughly. Which would be up here. So we're going to be basically creating a curve. Actually, it'll be more like this. It's going to, you know, kind of swoop down like this. Sorry, it's so like wobbly looking. But basically, this is what your curve would look like, that the smaller the fragment, the further the distance traveled. Larger fragments are going to get stuck in that agarose mesh. They're not going to get to go as far. And the principle here is basically, if you create this as your standard curve of, okay, this is our, you know, we've plotted out our ladder fragments and how far they traveled. Now we can take these fragments of unknown size. We don't know their sizes, but we know how far they traveled from their origin point. And so we can use their distance traveled to figure out their size in base pairs. So distance traveled, we find their distance traveled, and then we just go across and down and then approximate their size in base pairs. So very similar to what we did with the maltose concentration and absorbance. Pretty much like the same principle there, that we can use this standard curve to find the fragment size of an unknown fragment based on how far it traveled through a gel.
Professor, so we still need an equation, right?
No, you don't need an equation for this. This is just would be for, in this case, you're just like plotting it out. So you have your x-axis data, and distance travel would be y-axis. And then once you've plotted it out, you can just like use the graph and to kind of eyeball the fragment size. So, and also because it is like an exponential curve, it's not like a line of best fit, which is, you know, linear. When it, linear equation is much nicer to work with than the equation for this kind of curved or like sloped line would be. So in this case, you would just use the line on the graph to figure out the fragment size of our sample fragments, sample bands.
Okay.
So the distance migrated. So for creating this graph, the distance migrated, we figure it out by literally taking this ruler and measuring how far each of these ladder bands traveled from their origin, which is represented by this triangle, this little rectangle. So the way that this page was designed is that this 0 point lines up with these wells, and that's where our sample started. So as the electricity runs through, all of these, you know, fragments are getting separated. The larger fragments get caught closer towards the top of the gel. Smaller fragments keep traveling through. So this is where we get our data points to make that standard curve. So we have the DNA fragment size, distance traveled. And then we can figure out the distance that each of these little bands traveled. And if we know how far it traveled, and we have this nice curve, we can say, oh, okay, it went 32 millimeters. Go down here. Okay, so it's about 1,500 base pairs large. All right, so we don't need to spend any more time on this unless we still have questions.
Can you please scroll back up a little bit?
To this page.
Oh, I thought we did the box already. Never mind.
All right. Okay, so we'll just like let this simmer in your minds for a little bit. If you still have questions at the end, we can revisit this. So now we're moving on to the genotyping of these individuals. And we are imagining that we did this genotyping for another 2 families using the same PCR setup and the same restriction digest. So now it wants us to determine the genotypes and phenotypes for all the parents and children shown here in these 2 families, families A and B. So Kumari, to answer your question, this the standard curve for the migration distance, it's not going to be y equals mx plus b, because that is a linear equation, and this would not be a linear graph. It's going to be kind of curved. So, and that's why they don't ask you to use the equation in that case, is because the equation for like a curved exponential line is a lot more complicated than a linear equation. Okay, so we have our DNA ladder, then we've got our 2 parents and our 2 children. So based on these results, what can we surmise about these individuals? So what are their genotypes and what are their phenotypes? Oops. Maybe we can just remind ourselves up here. So if you have the HBA allele, what fragments would you find in your sample?
Two.
Right, so there'd be 2 fragments, and do we remember the sizes of those fragments? I think I have to look at my lab exercise because I don't have that memorized. So yes, there are 2 fragments. So we should have a fragment at 68 and one at 231. And then for the sickle cell allele, we do not have that restriction site, right? So we're only going to have the 1 fragment, which is 299. All right, so if we see 3 lines in someone's sample, what does that indicate?
Heterozygous.
Right, so they're going to have 1 copy of each allele. So it looks like parent 1 and parent 2 are both heterozygotes. And then what about the children here?
Homozygous.
Right, they are homozygous. Is it homozygous A or homozygous S? Well, they have the 2 bands And the only way you get two bands is if you have the A allele. So they're going to be homozygous for the normal hemoglobin allele. So we can put that up here for our answer. So, okay, in family A, parents are both HbA, HbS, so they're both carriers. And their children are both homozygous for the normal hemoglobin allele. Right, and then for family B, it looks like we have one heterozygote and then the rest are homozygous for the normal allele. That's good. So parent 2 and the two children, all three of these individuals are HbA, HbA. And just parent 1 is the carrier, or they have the sickle cell trait. All right, does everyone understand how we got those conclusions? If you don't understand, you should speak up now.
Professor, I don't understand that.
Okay. All right, so we have, let's look at family A. So these are people who, gave up a little DNA sample. We ran the PCR to, amplify just that segment of DNA with the possible mutation in it. And then we took that amplified DNA and digested it. But remember, the digestion only cut apart the normal HbA allele. And it did not cut the HbS allele because the cut site is exactly where that mutation is. So if the mutation is present, the fragment does not get cut. And that's what is reflected in these results, that if they only had fragments at this 299 base pair site, that would indicate the presence of this allele. If they have fragments in these other two locations, that indicates the presence of the normal allele. So an individual that has 3 fragments at 299, approximately 231, and 68 base pairs, that means that they are a heterozygote. So they have one of each of these alleles. So both of these parents are heterozygous, meaning they have the HbA, the normal allele, because they have fragments in those positions, but they also have that HbS allele because they have a fragment at that position as well. So they have one of each. And then their children each have 2 fragments. So child 1 has 2 fragments, child 2 also 2 fragments. And their fragments are in the position, the approximate position for this HbA allele. There's nothing in this 299 spot, so that tells us that they did not inherit that trait from either of their parents. So they are homozygous for the normal hemoglobin protein.
Okay, thank you.
You're welcome. Yeah, so it asks to explain your conclusions up here, so you can kind of summarize what I was just saying, that the so the mutant allele, the HBS, does not have a restriction site. So it does not get cut by the DDEI enzyme. So the wild type or the normal allele. Where'd my answer go? Okay. So I guess I ran out of space to write the answer, but yeah, basically that's the difference between having a cut site and not having a cut site. Is the same mutation that gives rise to this mutant protein also means that the fragment of DNA cannot get cut or digested by that enzyme.
How do we know like which line is the mutant?
So you would just have to know these values, or I guess even if you didn't have these numbers memorized, if you remember that the mutant fragment does not get cut, you can kind of assume that, okay, if it's not getting cut, then that's going to be the largest fragment in the sample. And the two smaller ones are going to be the fragments from actually being able to digest the main like target sequence.
I'm sorry, this might be like kind of dumb, but how do you know if it's cut or not?
I guess just look at the relative sizes. So I know it's kind of hard to estimate on here because it is kind of like arbitrary, that, you know, this is like created on a computer screen. It's not, and it's just meant to like vaguely represent the actual lab results. But basically you would say like, okay, the biggest fragment in this sample is this 300 band. And these other two fragments, they're obviously smaller because they're lower down on the gel. And, you know, okay, this one's a little above 200 and this is less than 100. So probably if I added these two fragment sizes together, it would equal this 300 one.
Okay.
And yeah, it just goes back to like, you have to remember, I guess, like the basic principles of this lab exercise, and the fact that the mutant one, the mutant allele does not get cut. So that's going to be the bigger band, or yeah, the larger fragment. And then the normal allele does get cut. So those would be the two smaller fragments.
Oh, I see what you mean. Okay.
So Camille, for your question, why can't we just tell if they have 3 or 2? So I don't know if your professor would accept that as an answer for this question, because it tells you it wants the genotypes and phenotypes. So if you just said parent 1 has 3 fragments, that's not a genotype and that's not a phenotype. So it's asking you for a very specific type of answer. I guess if you wanted to use that as your explanation, as like, okay, I determined the parents were carriers because there were 3 fragments in that sample instead of 2, that would probably be sufficient. But you want to make sure if it's asking for genotype and phenotype that you are giving that very specific information. But then, yeah, if you wanted to explain like, okay, why is that genotype, or how did you determine that genotype? You could say, oh, because there were 3 bands, there were 3 fragments instead of just 2 fragments, or instead of just 1 fragment.
And I'm sorry, how would you know if they were affected? We didn't finish our lab, obviously, so.
Oh no. Yeah, so the affected would just be, affected is if they are homozygous for the mutant allele. So if they were HBS, then they would be affected. And in that
Okay.
case, if they were homozygous for the HBS allele, they would just have one band and it would be here at the 300 mark.
Okay.
And they wouldn't have these other two because they don't have that allele.
Right. Okay. That makes sense.
Yep.
Thanks.
Yes, Annealing, to answer your question, one fragment would be the HBS, two fragments would be the HBA. So that's why these individuals here, they are HBA, that's the only allele they have. This person has all three bands. If there was just one band at the 300 level, that would indicate just the presence of the mutant allele.
So, Professor,
Mhm.
for us to see that the person is affected, there should be one solid fragment. Is that the indication?
Yes, you could.
If the person is affected, so how would it look like?
If it was, yeah, if they were affected, yes, it would be one solid fragment at the 300 level, because that would indicate that this would be the only allele that they have, that there's no HBA allele, just two copies of the HBS.
Okay, thanks.
Hey, Professor. So is this result is before enzyme reaction or after enzyme reaction?
This is after. Yeah, so this is after the enzyme because the enzyme is what cuts these fragments. So prior to cutting it, I'm going to draw it down here. Yeah, I'm going to draw it down here. Okay, so we have our like double-stranded DNA that this is our target sequence that we made copies of in PCR. And so this target sequence is 299 base pairs long. So if it is an HBA allele, it's going to have a cut site and that's going to be recognized by that enzyme. And so if it is HBA, it's going to get cut, let's say right here, and that's what's going to produce those two fragments, one that is 68 base pairs long and one that's 231 base pairs long. So if it is the mutant allele, it's not going to have that cut site. And so it will just remain 299 base pairs long. All right, so this would be an example of this HBA allele. And now let's draw our HBS allele. So again, we have our double-stranded DNA, that target sequence that we made copies of during PCR. Oh, but if that mutation is present, if it's an HBS allele, then there's no cut site. There's nothing for that enzyme to recognize to cut through the DNA. And that's why the fragment size remains at 299 base pairs.
Okay, thank you.
Yep. All right, so then this question asks us, it says another couple was screened for the trait and they were found to have the same banding pattern as the parents in family B. All right, family B, we had one parent who was heterozygous and then one parent who was homozygous normal or unaffected. All right, so we're keeping in mind these two parents. It's asking what is the chance of any of the children being affected with sickle cell disease or sickle cell trait?
There's no chance, right?
No chance for the disease, but they could still be a carrier.
Okay.
So we can draw, we can actually draw a little Punnett square for this. So if, oops. All right, so if one parent is AS, we'll put that there. So one parent is A. Oh, this isn't working. Okay. One parent is AS. The other parent is AA. And then we can just complete our Punnett square. So AA. Oops. Okay, so there is, 0% chance of having sickle cell disease. But a 50% chance of at least having the trait. The sickle cell trait is 50%, and then 50% of not being a carrier at all. All right, so let's move on. I think that might be the last
Can
for.
I do one more, maybe the notes. Okay, let me get.
Mhm.
Thank you.
All right, so it looks like we are moving now to exercise 9 and then finally exercise 8, which exercise 8 is super easy because that's just about mitosis and like identifying, different phases of mitosis. So, all right, bacterial transformation. So this was the exercise where you were isolating that plasmid and then putting it into these competent cells and testing whether or not they had taken up the plasmid by making them grow on agar with ampicillin in it. So this first part is just matching these terms, just so you know what they are. So what is LB agar? I don't think the matching like this directly is going to be on the exam, but you will need to know what these are because there might be, I think there are some like free response questions where it asks you like, what is a plasmid or what does competence mean or like what is pGlow? So you'll want to know what each of these terms are.
Two.
Yeah, LB agar, that is the that's our nutrient stuff. All right, what about glycerol?
That's 5, I think.
Yep, yeah, that's going to be our cryopreservative. All right, ampicillin.
That's 6.
Yes, ma'am. That is our antibiotic, amp R gene.
Is it 12?
Calcium chloride.
3.
Yeah. Beta lactamase.
7.
Right, our plasmid.
1.
pGlow.
9.
Competence.
4.
All right, transformation.
8.
Yes, ma'am. Amp R E. coli.
10.
Yep. And then amp S. E. coli.
11.
Yeah, process of elimination. Yeah, so as long as you know what these terms are and also like what their importance was in that experiment, you're in good shape. there will be a section on the test where they'll give you kind of the results of a transformation experiment, and you have to interpret those results. So you have to know like what it means if there's growth on a plate or if there isn't growth on a plate, and have to like explain that. Go over D and B. Yes. All right, so glycerol. Glycerol is just this molecule that is, or it's a reagent kind of, that's it's used to freeze the cells. So it's a way that you can freeze cells without damaging cells, because normally, you know, cells are mostly water. So if you just freeze a cell, the water creates little like tiny crystals that will basically stab through the plasma membrane. And then when you thaw those cells out and those ice crystals dissolve, what you're left with is like shreds of a cell. And if you've ever frozen meat, like let's say you buy a big pack of chicken at Costco and you freeze some of it, when you take it out and thaw it, the texture is a little bit different. It's not quite the same as when it was fresh. And let's say you kept taking that same piece of chicken and freezing it again, and then thawing it again, and freezing it again, and thawing it again. That chicken is just going to get like less and less desirable to eat, because it's just going to completely like lose its texture. And it's because you have these microscopic ice crystals that stab through the cell membranes, and that's why it changes like the texture of meat. So glycerol prevents the formation of those crystals, and that's why it's a good cryopreservatives. And then D is the AMPR gene. So this is the gene that, well, the gene is a sequence of DNA, right? And a sequence of DNA is going to code for a protein. So the AMPR gene is going to code for the creation of this enzyme called beta-lactamase. And the beta-lactamase is actually what breaks down ampicillin and makes it useless. So any bacterial cell that has the AMPR gene is going to be able to grow in the presence of ampicillin. The ampicillin is not going to kill it because it has a way to break up that ampicillin and render it useless. Any other terms on this list we want to go over really quick?
Can you go over P-glow?
Yeah. So P-glow, this is the name of the plasmid. And a plasmid is this like extra piece of DNA. It's not part of the bacterial genome. It's just an extra bit of DNA that, you know, bacteria can take up plasmids from their environment, or they can get them from their bacterial cell friends. And so P-glow is just the name of this plasmid that we used that contains the ampicillin resistance gene, as well as that gene for actually fluorescing and glowing under a black light. All right, any other ones? So these are two videos. I don't know why the links are black boxes. It's kind of weird, but yeah, if you have this file and you click on these black boxes, it'll open up these YouTube videos. You can also reach them by clicking these links here, but it just kind of walks you through like the principles of bacterial transformation. This has a nice little like visualization of the process. All right, so here we have some kind of some concept questions about the experiment. So this alludes to kind of what I was just talking about. So what is the link between P-glow and ampicillin resistance? So P-glow is the plasmid. This is a piece of DNA that carries genes on it. So it carries genes, including a gene called AMPR. And what is special about AMPR? What does it create? So AMPR is the sequence. That sequence is going to make a protein. What is the protein that AMPR makes?
Is it the green fluorescent pigment?
So that is one of the genes on pGlow, but that's not what AMPR makes.
Lactamase?
Yes, AMPR makes the protein beta lactamase, which is that enzyme that breaks apart ampicillin. This enzyme digests ampicillin and makes it useless against bacteria. I guess we could expand on this a little bit and say any bacterium that has taken up the pGlow plasmid is resistant to ampicillin. All right, so how do we know that the transformation worked? There's like two ways that we could tell that the bacteria were transformed, meaning they took up that pGlow plasmid.
It's glowing, I think so, with PFG, I think.
Yes, that's one indication, is the glowing. We know that the transformation was
And the dots?
The colonies or whatever.
the colonies on which plate?
Oh, no, I don't know.
Okay, well, you're on the right track there. So we know the transformation was successful if the, if there are colonies growing in the presence of an antibiotic and the colonies glow. Light. So transformation means they took up this plasmid, which contains the AMPR gene. If they have that AMPR gene, they're going to be able to grow in the presence of what?
Ampicillin.
Right, yeah, ampicillin, exactly. So the transformed bacteria, we grew them in agar that had ampicillin added to it. So normal bacteria can't grow in the presence of ampicillin. There's no colonies there. It's completely bare. But if they have the pGlow plasmid, if they took it up and were transformed, that means they're going to grow. We're going to see colonies on that ampicillin plate, and the colonies are going to glow under UV light. Okay, so what kind of controls did we use in that transformation experiment? So remember, we had like the competent cells that we added pGlow to, and then what did we add to the other cells?
Could you repeat that? I think, well, we had a negative and positive control, and those were B and C.
Right.
I think B was the positive control and C was the negative control.
Okay, yeah, so we had plate B was the positive control. And what was that positive control like? What did we add to it, and what was like it growing in that plate?
There was a lot of bacteria. We added, forgot, it was the agar, and there was something else. I forgot what was.
Well, yeah, so the positive control was where you had like the carpet of growth, right? Or there was, I forget, when was it no growth? But yeah, so there was a carpet of growth because it was just agar with no ampicillin added, right?
Oh wait, maybe it was C that was the carpet of growth.
That's okay.
Yeah.
Okay. Right, so the negative control was agar with ampicillin. And we had no growth on that one, right? No. You're right, and our positive control was just agar alone. And then we had, what do we call that? A lawn of bacteria. Right, and remember that for both of these plates, we added E. coli that did not have any P glow in it, right? It was just E. coli, and I think we added a little bit of water, but there wasn't any special transformation there. It was just plain E. coli. As we can say, plain E. coli on agar with ampicillin.
Yeah, one was like a lawn of growth and one was like no growth at all.
Right.
Yeah.
Yeah, so what does each control tell us when we compare them to plate A? Agar is just the growth medium. It's just that like stiff gelatin stuff in the bottom of the plate. It just gives a place for the bacteria to grow, and it provides them like nutrients and stuff that they like to make sure that they grow. That's all it is. So what was the benefit of having this negative control? Right, so we had plate A with our little glowing colonies, and then plate B had no growth at all. So what was the point of that? What did that show us? Excellent, Diego. Yeah, it showed that the E. coli we were using were not already resistant to ampicillin, that they were susceptible to it. And it also confirmed to us that the ampicillin we were using was effective, right? It hadn't like expired or been somehow like insufficient or something. Like the ampicillin that was used in the experiment was effective at what it was supposed to do. It was able to prevent bacterial growth. All right, and then plate C. What did plate C show us? So we had plain E. coli on regular agar, and it was able to grow. So what does that demonstrate?
Elsa,
Mhm.
we see like when we did the experience in the lab, we saw that on the plate C, there were like a go, there were some bacteria.
Right. So the fact that the bacteria was able to grow on plate C showed that the bacteria was viable, and yeah, it could flourish if you gave it the right conditions. So both of these controls had a purpose because plate C showed us that yes, that our E. coli is alive and thriving. If you give it the right conditions, it'll go to town. And plate B showed us that our ampicillin is effective and does what it's supposed to do. So that just strengthens our case for the transformed bacteria in plate A, that these ampicillin that normally would die survived, and they also glow, which is just a fun party trick. So yeah, so plate C showed that the E. coli was viable and could grow in the right conditions. All right, and just to reiterate once again, what is it about pea glow that allows the bacteria to survive? What does the pea glow have that confers this survival or this resistance for the bacteria?
They're able to destroy it using the beta lactamase.
Yes, exactly. If they take up the pea glow plasmid, they then have that special AMP-R gene, which creates beta lactamase. Oops, because they AMP-R gene creates beta lactamase. And allows them to destroy ampicillin. All right, I'm going to scroll down a little bit. I'll try to keep these answers up here. So this, I distinctly remember seeing this on the exam that I looked at. So there will be a question where they'll present you with results of this kind of transformation experiment, and you should be able to interpret what those results mean. So for example, here it's showing you 3 different plates, what the plates have on them, and then what was added, what kind of cells were added to each plate. So you should be able to predict what the growth pattern would be on each of these 3 plates, and also explain why you would see that growth pattern. All right, so we have this first one, we have LB agar, ampicillin, and arabinose plated with the E. coli plus the pea glow. So let's see, we got a little drop down menu here with tiny text. Okay, would we expect to see a bacterial lawn, no growth, colonies of ampicillin susceptible E. coli, colonies of ampicillin resistant E. coli, or a lawn of ampicillin susceptible E. coli?
Colonies of ampicillin resistant E. coli.
Yes, absolutely. Yeah, we would have colonies, not a lawn, and those colonies would be resistant, ampicillin resistant. All right, the next one is agar plus ampicillin, and we're plating it with just E. coli and water. So what would we expect to see there? No growth. Yep. Yeah, so we would have nothing there. We wouldn't have a lawn. We wouldn't have colonies. There's just nothing. It's desolate. And then lastly, LB agar plated with E. coli and water.
First one.
Yeah, so actually, I guess there's two possible correct answers. You could say bacterial lawn of E. coli, that would be my instinct, but then they also have lawn of ampicillin susceptible E. coli, which I guess would also technically be correct. If I was grading this exam, I would accept both those answers. So yeah, you definitely have a lawn on this one. No growth in the middle here, and then little glowy colonies over on the first plate.
I do have to say, when we did our experiment, we had a beautiful lawn of glowy E. coli.
Oh, really?
Yeah, we
Oh.
spread them out really well.
Okay. Nice. So they were like super hardy and resistant.
Yes, they were very pretty.
Yeah, it's fun to make stuff glow. Feels like you feel like a mad scientist or something. Like, ooh. All right, I'm going to scroll on down. Okay, and so finally we are here to mitosis. I believe this is, let me see how many pages we have left for this. Okay, so we've got some identification of phases. Oh, and then a blank page. All right, so let's just get through this and then we can kind of wrap up. Of course, I mean, you're always free to leave at any time. You don't have to stay, but yeah, so we're almost done here. All right, so what are the 4 stages of the cell cycle? So not mitosis, but the cell cycle. And what's the primary activity in each of those stages?
G1, S, G2. Is that what you mean?
Yes, yeah. So we've got G1.
For growth.
Yeah.
Okay, sorry, yeah.
Oh yeah, growth.
S for, what is it? S, making the protein, oh, not the protein, synthesis.
Yeah, what are we synthesizing here?
Good question.
You are right that the S is for synthesis. Right, that's going to be our DNA replication. So DNA
Okay.
synthesis. So we are replicating our whole genome, getting ready for mitosis. All right, then we have our G2.
Getting growth again, but getting ready for mitosis.
Yeah. Yeah, more growth, prepping for mitosis. Yeah, and then finally M is our actual division phase. All right, how do we differentiate between mitosis and cytokinesis?
Splitting of the
stage.
cytoplasm.
Yeah, so mitosis is splitting of something. Cytokinesis is splitting of the cytoplasm.
Yeah.
What are we splitting in mitosis?
The chromatids.
Yeah. Yeah, I would say chromosomes or genetic material. You can say anything like, yeah, chromosomes, genetic material. You could say nucleus if you wanted to. Yeah, but you're dividing up the genetic goods during mitosis, and then cytokinesis is splitting up like the cytoplasm and assuming also the organelles and stuff, everything gets divvied up. All right, how does this process differ in plant and animal cells?
Animals make a cleavage, whatever it's called, furrow or something. I don't remember.
Yeah, animal cells form that cleavage furrow. And then what do the plant cells do? So it's a precursor to the cell wall.
Oh, okay.
Cell wall kind of answers the second question, why don't plant cells undergo cytokinesis the same way? Plant cells have a cell wall. So what do we call that, like, formation of the separation between two plant cells? So we call that a cell plate. So the cell plate is kind of the precursor to the cell wall that will, like, ultimately separate those two new cells. Okay, so knowing the number of chromosomes and the physical state of chromosomes in each stage, really what it's going to get at, there might be a question or two, and I think these are usually like in the multiple choice question section, where it'll ask you about, you know, if a cell has a given number of chromosomes at this phase, how many does it have at like some other phase in the cycle? So let's walk through this really quickly so we understand the concept here. Okay, so we're talking about human cells that have 46 chromosomes at G1. So G1, there are 46 chromosomes. I'm going to abbreviate chromosomes. How many is it going to have? Well, and then in S, we're going to copy. So how many is it going to have in G2?
92.
Right. Yeah, in G2, we're going to have 92 chromosomes because we've made a duplicate copy of all of them. And these chromosomes are going to be conjoined, right? So we're going to have 92 chromosomes as 46 conjoined sister chromatids.
I'm sorry, I have a question.
Yeah.
Are we going to say 92 chromosome or chromatid?
I think you could say either. As long as you said sister chromatids, then I think it would be implied that you know that they've been duplicated and they're still like conjoined.
Okay, thank you.
So I think either of these answers would be correct, 92 or 46. Okay, so that's at G2, and then we're going to go through phases of mitosis. So let's say at P phase, we're still going to have our 92 chromosomes or our 46 sister chromatids. And then what about in M phase? Still the same thing, right? Except now they're lined up in the middle. All right, what happens in anaphase when we separate those chromatids?
They split up?
Yeah, so this could be, this you would express by saying 92 single chromatids, or yeah. Because they're no longer sisters, you've separated the sisters, and so now instead of the 46 sister chromatids, you would have 92 single chromatids. And then our telophase, you could say single chromosomes. Now we're back to having 46 chromosomes like where we started from. And this is really just, this kind of question is really just to demonstrate that like you understand what the arrangement of chromosomes is at each phase of mitosis, that you know when the DNA has been replicated, you know when the chromatids are still together and when they are pulled apart. So it's just a different way to demonstrate that you understand like what is happening in each phase of mitosis. Okay, so why, when we were observing the phases of mitosis, why did we use these two cell samples, the onion root tip and the whitefish blastula?
They're constantly dividing.
Right, yeah. Constantly dividing. What are the genetic consequences of mitotic cell division for the resulting daughter cells? So what are the daughter cells for mitosis genetically like? Are they similar? Are they identical? Are they different?
Identical.
Identical.
Yeah. Mhm. Yeah, they are exact replicas of each other. They're clones of each other. Oh my gosh. Okay, and so why do we need mitotic cell division? As multicellular organisms, what does it do for us?
For our growth, or if you have like an injury, to make cells again.
Right, so for tissue
Repair them.
repair or injury repair, development, because obviously you're a different size now than when you were a baby. Yeah, so repair, growth, development, basically those three things. All right. And now we're moving on to this phase. And this is a little bit annoying because these pictures are so tiny, but we'll try our best. Okay, let's start with an easy one. So this one up here, what phase is this?
Metaphase? Oh, no, anaphase?
Yeah, so that would be anaphase. So we can see how they're being pulled apart. Let's do this one over here. Yeah, I would say that's pro-meta or even metaphase because it kind of looks like they are lined up. That's definitely a lateral view. What about this one? So we can clearly see the chromosomes, they've condensed, but they're not quite lined up yet.
Prophase?
Yeah, so that would definitely be like a pro-metaphase. So they're on their way to lining up like this, but they're not quite there yet. All right. What about this one here? All right, so we have a vote for anaphase. I'm not quite convinced of that because they don't really look like they're coming apart. But we definitely have condensing of the chromosomes.
Prophase?
Yeah, I would say that's prophase. What about this one up here? So this one,
Telophase?
not
No, never mind.
telophase.
I'm
Yeah.
looking at the wrong one.
It's all good. Yeah, so we can't quite see the individual chromosomes yet. They're not quite like fully condensed. So I would say this is interphase. Whenever you can like clearly see the nucleus, and it's like you can even see how this genetic material is still kind of contained within a membrane. So that would indicate that we're not quite at prophase yet because we haven't dissolved that membrane. Okay, what about this guy up here? I think it's indicating like these two like little tiny mini cells.
Cytokinesis.
Cytokinesis.
Yeah, exactly. That would be a cytokinesis. And you can kind of tell the nuclear membrane is reformed again. What about this one over here?
Where's your mouse? I'm sorry.
Oh, right here.
Okay.
Yeah, I would say this is also like a telophase cytokinesis. Some of these are harder to tell than others. I know that whenever they have the exam ones, they make it pretty obvious. It's not, they don't leave it like ambiguous cells. All right, what about this one over here?
Maybe metaphase.
Yeah, this one is hard to tell because obviously the chromosomes have condensed. So it's like, okay, we're past prophase, but they're not quite like aligned anywhere. Yeah, so maybe like a prometaphase or something. They're looking a little messy there, but all right. The point here is just as long as you can tell like what phase these cells are in, you're in good shape. I know some people get confused by interphase, that like they'll mistake this for prophase, but you just have to keep in mind that prophase, you should already see these individual chromosomes. You should see these strands. If it still looks like a big heap of like spaghetti or just these, dots, and you can't tell the difference between the chromosomes, then it's an interphase. And also being able to see kind of like the outline of a nucleus would also indicate you're not in prophase yet, you're still interphase. All right, let's move on to the whitefish down here. Okay, let's do this one here. Yeah, that looks like a nice metaphase. And here we have to keep in mind because, we're looking at this three-dimensional ball of cells. We could see metaphase from the lateral view or from the polar view. So this would be metaphase from the lateral or side view. If we looked at metaphase from like a top-down view, it would look like this. So everything's still arranged in the middle. We just have a different perspective of it. So this would be an example of metaphase from a polar view. So instead of looking directly at it from the side, you're looking down on it.
Do we need to know that for the test? Are they going to do it that way, or?
They might, yeah.
Okay.
But you really only see it that way in the like mammalian cells, or not mammalian, animal cells.
For the polar?
Right, yeah.
Okay.
Because plant cells, everything is so like rigid and kind of like lined up, you really only ever see it from one perspective. So that's something you only have to worry about in animal cells.
I'm so sorry to interrupt, but is it possible that you can scroll up for 2 seconds so I can look at the top thing really quick? Thank you. That's it. Thank you.
Okay. Yeah, and I see from the time that we're approaching 9 o'clock, I'm still gonna stick around just to answer any final questions. Like I'm not in any hurry to leave. If you are, that's okay. And I'll be making this recording into a YouTube video and then sending the link out to your instructors. Yeah, we'll do it some more.
I have a question. Will you, when we're finished with this, will you send out the video like right after or like later today?
It'll be later tonight. Like it usually takes a while to convert it into like a YouTube file. But as soon as I have it made and I do that link, then I can send that out.
Okay.
And if you really don't trust your professors to send it out and you want me to send it to you directly, then I'm going to put my email in the chat. So email me directly if you want like a direct link.
I was going to ask you that. Thank you.
Yeah. So my email address is really easy. It's just @Montgomery College. But all right, yeah, let's do a few more of these whitefish ones.
Would that be cytokinesis? Maybe telophase?
This one, so I would say this is still anaphase. It's late anaphase because you can see how far apart these are. What would indicate like cytokinesis would be you would already start to see like a line here or like a pinching in between the two cells.
Okay.
Yeah, so I would say this is anaphase. Yeah, and this would be like a telophase cytokinesis over here. So this one up here, where I know the arrow is pointing to this little globule, don't pay attention to that, but here's the nucleus, and you can see that like these, the genetic material isn't fully condensed yet. So this would be kind of a pre-mitosis. I'd say this is either interphase or prophase. Oh yeah, actually this would be prophase. You see over here where you can't even tell like where the nucleus is really, or like you don't even see any, it looks like it's an empty cell basically. This is what interphase looks like in animal cells. So it's like, it really looks like there's nothing going on. And then once those chromosomes start to condense, then they really come into view. And so that would be your prophase. This would be your interphase. Let's see, this would also be, here's another metaphase. And here's another good anaphase where I feel like anaphases are pretty distinct look to it because you can clearly see like those two groups of chromatids being pulled apart.
So for like the top right, would you say that's anaphase?
Yeah, this is anaphase. Yeah, because like if you look at, you can kind of tell where the boundary of the cell is and you don't really see any like pinching in yet. Yeah, so it's definitely still anaphase. Yeah, it doesn't even look like there's too many good telophase ones. I think this is like the only prime example of that, where you start to see, first of all, you can see that little pinching in, that little divot there, and then also the formation of that cell membrane.
Professor,
yes,
what's the difference from the metaphase and metaphase polar view and anaphase?
So the polar view, so you can still see, you can still kind of identify it as metaphase because all of these chromosomes look like they're in the middle. And, you know, in anaphase, they're already starting to be pulled apart. So like this one up here, for example, I would also say that this is a metaphase from the polar view. And your textbook should have a good image of that too. Like basically the polar view is, it's like if you had a circle and then these squiggly lines or like these little V shapes, and the point of the V is like pointing inwards, like towards the center, but all of the chromosomes are still like in the middle of the cell. And it's like really when you start to see them pulled apart and like in these two distinct formations, that's really your anaphase there. So you can see like these are all clumped together. They're not separating. So that's definitely a metaphase. And then once they start creating that distance in between them, yeah, the top left one, this would also be an anaphase. Yeah, you can clearly see that.
Okay.
They have that distance. Yeah, these two would also be metaphases. So this would be a metaphase from the lateral view. This would be a metaphase from the polar view. What do we think about this cell here?
Prophase?
Yeah, that would be a prophase. So it's not quite condensed enough to be a metaphase. It does sort of resemble the metaphase from a polar view, but when you're looking at these two side by side, you can see how much like sharper that definition is here for those individual chromosomes. Whereas here, it's still kind of formulating. All right, I think that is the end. Yeah, that's it for this review. So thank you so much for attending and for participating. And oh, actually, Hawaii, that is a great question. I do have some information there. I jotted it down about the exam. So I'm going to scroll down a little bit. Yeah, you're welcome. And yeah, like I said, if you want me to send you the YouTube video link directly, just email me and I can do that for you. But okay, the exam. Oh, here it is. All right, so there's going to be, it starts with 20 multiple choice questions that are worth 2.5 points each. If you didn't have to build graphs on your first exam, you probably won't have to on this one either. You'll probably just have to like interpret graphs and interpret results as like part of the free response. So there are 12 free response questions, and those point values vary. So some of them are only worth 4 points each, some are worth 5, 6, 8, and some are worth 10. And most of these, most of the free response questions are like multi-part. So even though there's only 12 of them, sometimes they'll take up like a half a page or a page because there's like several like parts to the question. So this first part is worth 50 points total. And then the second part, the free response part, is worth 70 points total. And Julia, to answer your question, does each professor make their own exam? So the course coordinator kind of sets the standards. That's Dr. Castanos. So she'll create like the basic exam, and we are allowed to alter it slightly, like to our own needs, or like if we had, if we emphasize certain things in our lab exercises, like we can add questions on that, or like take out questions on things that like we didn't touch on as much. So we do, like, each professor does have some like autonomy to create their own exam, but the basic exam is still the same. So your professor, maybe they added a few multiple choice questions so that there'd be 2 points each or something, or maybe they add extra credit and someone else doesn't. So they're it could differ slightly between professors.